Okay, so welcome to the next Google Hangout hosted by MS Translate. This is our third Google Hangout as part of our involvement with the MS team at the Melbourne Brain Centre. And tonight we have the pleasure of bringing you Dr. Melissa Gresle and Gerry, who are both members of the basic science team at the Melbourne Brain Centre. And over the past couple of days, you will have seen some summaries of their research dealing with some of the genetics of multiple sclerosis. So the first thing that we're going to do is hand over to Mel and Gerry, and they'll give a brief introduction as to who they are and the research that they do, and then we'll throw it over to you guys to ask them some questions. So Mel, do you want to start us off?
Yeah, sure. So as Brett mentioned, my name is Melissa Gresle. I work with Helmut Butzgugen, who hopefully you've seen the video that he's put on the MS Translate Facebook page earlier in the week, explaining a little bit about some of the work that we do. And our lab is based here at the Royal Melbourne Hospital, mainly because the work that we do is based looking at blood samples from people with MS. So the main areas of research that we're interested in are understanding MS genetics and also looking at ways to measure injury in people with MS, and possibly also understanding mechanisms of action of some of the new oral therapies for MS. So Helmut's introduced some of the genetics work that we do, so I won't talk much about that. But the other side of the research that we do actually involves looking at developing new blood tests for measuring nerve fiber damage in people with MS. And the reason that we're doing this is that we now understand that damage to nerve fibers is really important in driving disability in people with MS. And at the moment, we don't really have a good way of actually measuring this and monitoring what's happening in people with MS. So we decided that we would work in collaboration with a scientist out at the University of Florida, Gerry, to see whether we could use his blood test to measure nerve fiber damage in people with MS. As you saw on the website earlier in the week, we're able to show that yes, it is possible to detect fragments of broken nerves in the blood of people with MS. We found that levels of these nerve fragments or proteins in the blood were associated with worsened disability and larger lesions on MRI scans. And hopefully the idea is that we might be able to develop these kinds of tests to perhaps monitor what's happening in people with MS, and perhaps more importantly to test how effective drug therapies are at modifying nerve fiber damage, and also perhaps testing new kinds of therapies against this type of damage. And the other project that we're working on at the moment is trying to investigate how new oral drug therapies for multiple sclerosis, so Gilenya and Tecfidera, might actually work to modify the processes that are occurring in immune cells, so to understand how they're actually working. And the reason that we're doing this is, although they're used clinically, we don't fully understand how these drugs work. So we use a method known as gene expression microarray to look at how these drugs change levels of genes in immune cells to see if we can figure out what they're actually doing, so what pathways are they modifying in these cells, and can we use this information then to start to design newer kinds of drug therapies that are more specifically targeted at these pathways. And this would be important as it would help us to sort of reduce the side effects associated with sort of unwanted effects of these kinds of drugs, and might also help us to develop newer kinds of drugs that are more effective. So, yep, that's my little introduction there, so I'll hand over to Gerry.
Hi, everyone. My name's Gerry. I'm a PhD student at the Melbourne Brain Centre in Parkville. I work with Professor Trevor Kilpatrick and Michelle Binder and Dr. Judith Field. And basically, our main focus of research is on a family of proteins called the TAM receptors. So I did my honours year in the lab looking at one of the members of this family of proteins and its role in a mouse model of central demyelination of MS. And subsequently, my PhD work has taken me into looking at genetic associations in some of these in some of the genes encoding for these proteins in MS, and also further looking at some more mouse work using mouse models to look at what exact roles these proteins are playing during demyelination processes, including modulation of the immune system and also potentially promoting the survival of some of the cells that are important in MS and demyelination. And also, more recently, some of my work has been looking at whether the levels of some of these molecules in our blood can be associated with either severity of MS or whether they can be kind of a biomarker for MS, or whether they can be used for stratifying patients based on what kind of disease level they may have. So that's a basic summary of the research that I'm doing.
Okay, well, we'll hand it over for questions now. Thanks, guys. Maybe one of the first questions that I'll ask, because it sort of follows on from what you were just talking about there, Gerry. It's come from Helen, who's going to be typing her questions through for us. Mm-hmm. And she's the question that she's asked, to sort of put it in, I guess, a question format, is that mice don't get MS, so how the demyelination in mice is not MS. And I guess the question that she's sort of asking about that is the mouse models that we use for MS, how applicable are they to the human disease, and how good are those models in terms of when we want to try and translate anything we see across to the human disease?
Mm-hmm. Yeah, that's a really good question. So basically, for one thing, because we don't know what the exact cause of MS is, there, I guess, isn't really a good way of causing the disease in experimental animals. So what we do have are experimental models where we can model certain parts of the disease. So, for example, a couple of the most commonly used models that I work with, and a lot of MS researchers work with, one probably know in the human disease in MS, the myelinating cells, or the oligodendrocytes in the central nervous system, are lost, and this is what causes the demyelination of neurons. So in this toxic model, we can give a toxin that specifically kills these oligodendrocyte cells, or the myelinating cells, and this is one way for us to model the loss of oligodendrocytes. However, as you may also know, MS is also an inflammatory disease and involves the immune system as well. And this toxic model doesn't model that immune response. So we have another model which is basically an autoimmune model of MS. So here we're causing an autoimmune attack of the central nervous system of the mice. And this models that particular aspect of MS. So because we don't know the exact cause of MS, we can only model certain aspects of the disease, and we have specific tools that we can use to model certain parts of the disease. But obviously we can't model the exact same disease in experimental animals as we do in humans. So depending on what the outcome measure is, I guess you would pick the model in which you would want to, which best represents the outcome that you want to measure in the end. Does that answer your question? Yep,
excellent. We got the thumbs up. Helen's got another question, but I will ask that in a second. Tony, do you have any questions that you'd like to ask? Tony or Brad?
Are we good at the moment?
Good at the moment. Annika, have you got any questions that you'd like to ask along?
I guess not really a question. I guess just to add to what Gerry said about the mouse models and things. I mean, I'm pretty much a clinical researcher, but I think the mouse models and the whole experimental encephalomyelitis model in mice has certainly advanced the science quite dramatically. Although it's clearly not a perfect model by any means. but I think the proof of the pudding is always in the human studies. But without those basic studies being done in animals, we just wouldn't be able to do, human studies. It's just unfortunate that the animals have to suffer for all of us to gain, because, there's a lot of stuff you can legally do to animals which you can't do in people. So, we have to start somewhere, don't we, to try and understand where to take the next step to. And I think a lot of the actual therapies that has been developed is, is on the back of, lots of mice that's been killed. So, it's a good starting point, but yeah, not perfect.
Yeah, Annika's absolutely right. The studies that we did with our assay actually started off in mice, and the reason that we had to start in mice is we needed to prove that what we were actually measuring was what we thought, so a damaged nerve fiber product. And to do this, you need to compare what's happening in the blood with what's happening in the brain or spinal cord, and obviously you can't do that in people. So often we start our experiments just to prove our initial hypothesis and idea before we can then move them into humans. So although, like Gerry says, it's not MS, there are similarities, and we can use those tools to start developing and testing our ideas before we move them into humans.
Yeah, and I think a lot of the time, Helen just posted a comment here about a study done in dogs in 1935 on chronic venous obstruction. I mean, clearly, Helen is from CCSVI Australia, so she clearly has a specific interest and agenda. But, this is, I guess, from my point of view, one of the things where the mouse model certainly has not shown any CCSVI issues, and neither has the human studies so far. So it's still a bit of progress that has to be made in that field.
Well, I'll bring up Tony. Do you have a question, or otherwise I'll ask a question?
Brad might have a question.
Brad has a question. Yeah, go for it, Brad.
Yep. It just came to mind. I just wanted to do the two mouse models spoken about, the inflammatory and the toxic model. Are they able to reproduce the localized myelin damage that you see in MS, or do they provide a uniform myelin damage in the mice?
Yep. So in the toxic model of demyelination, that particular model, there's specific demyelination in only certain parts of the brain. So specifically the bigger white matter tracts. And what we see particularly is that there's demyelination in the toxic model in the corpus callosum, which is the structure that connects both hemispheres of the brain. And then in the other model, the inflammatory model of MS, that model is more of a spinal cord model. So there you mainly see lesions in the spinal cord of those animals. So focal lesions in the spinal cords of those animals.
Yep. Okay.
Does that answer your question, Brad?
Yeah, I'm just interested. What exactly do you use to cause that inflammation to generate that response?
Yeah. So basically, so it's working, I guess, on the principle that of the autoimmune attack of the central nervous system. So basically, there we immunize the mice with a protein that's found in myelin, and we give that in what's called an adjuvant. So something that stimulates the immune system, and then that basically the adjuvant and the protein basically causes the animal's immune system to attack that particular protein in its own central nervous system. So that's how it's induced in the experimental animals. Does that make any sense?
Yeah.
I guess one of the most interesting things about that, Gerry, is that we can actually induce that with a lot of different myelin targets, can't we?
That's right.
Not that there's just one target that we can inject in, so sort of like myelin basic protein is one of the ones that's
right.
talking about. But there's a number of other targets in the myelin sheath that we can induce that disease with.
Yeah, that's right. So, and also, yeah, as Brett mentioned, yes, even depending on the different protein that you induce, it actually produces a different kind of disease in the mice. So, for example, you can have the relapsing remitting, or kind of relapsing and remitting model, or kind of a secondary progressive model, and so on as well. So, depending on the different peptide, you get a different kind of disease course in those animals.
So I might go on to Helen's other question that she asked previously. She was wondering if either of you would be able to comment on a publication that came out recently about polymorphisms in the gene coding for iron binding and transporting proteins and their association with disability severity and early progression in MS. You guys aware of this publication and able to comment on it?
I had a quick look at the manuscript before joining you guys. Yeah, it's an interesting paper. It's one of these studies where they seem to be looking at whether you can use, genetic variations to predict whether somebody will have a worsened disease course. And this is an idea that's sort of been thrown around by large companies like 23andMe. You heard of that one? So there's people trying to set up these biomarker panels to look for ways to predict whether people will develop a disease, and this one seems to be a study that's looking whether you can use these to predict whether people with MS will have a worsened outcome. And these kinds of studies are important. They fit, if you can prove these, yes, they're useful clinically, but of course it's going to be very difficult to just use this one indicator. It's going to be something that will be used sort of with other information to determine the likelihood of someone progressing. And also, the other side of this is it doesn't provide any real-time information about what's actually happening in a person with MS. So you'd really need to start to develop other kinds of measures to really see what's happening and how people are responding to their drugs. So it seems to be more of an indicator rather than something that can be used sort of in real time to monitor people. And that was my understanding of that study. I don't know if there was a specific question that you had about that.
No, I think it was just the general sort of comments. Does that sort of answer what you were asking, Helen? Actually, I don't know that you can hear me.
Does that answer your question, Helen? Brett asked.
We can't hear Helen.
No, we can't either.
No, sorry.
If she types her comment, we'll move on. You can just tell her to type her comment and we'll move on to another question.
Okay.
I was also like, I saw Helen's question and I tried to pull up that paper. I guess, for me as a clinician, when I look at any genetic study of any sort of association, the first thing I look at is how many people were in the study. And, you kind of always wonder, because, what would you think is the biggest things that people should be watching for when they see these news reports about this gene and that gene, and okay, now it's all linked to this certain thing that's been found in a lab somewhere. Like, what sort of caution would you think people should apply when they hear these sort of things? Because it doesn't always mean that it's causation. If there's a correlation, it doesn't mean that's the cause. And we have to be so careful because there's so much research and there's so many studies, and everyone has doing these things with the best of intent. But sometimes, you can get so lost with all this information. So I don't know if you guys have any sort of comments that you would say if you hear these reports. What would be your sort of red flags that you would think, oh, this should be taken seriously and this shouldn't?
Yeah, I mean, I think you're absolutely right, Annika. That there are definitely like heaps and heaps of these studies being conducted, and associations with MS popping up in lots of them. I think mainly the main thing that I guess we would look for is that if there is an association found in a gene coding for a specific protein, then I guess we would usually look and see if that protein has been previously implicated in some process that's actually important in the demyelination or in a particular animal model of MS. Whereas if it's just something that's just completely out of the blue and random, then I guess that adds to your point where it's simply correlation and not causation. And with such a complex disease as MS, it's not going to be one particular thing that causes the disease. Much rather, it's probably more of a combination of risk factors rather than just one particular thing. So really, you have to, as you say, be very, very careful when you're looking at these studies, and to, I guess, have you need kind of an educated, I guess, view of how to look at them, and I guess basic background on whether these genes are actually involved in some of the processes that are actually important in demyelination or MS.
And I mean, I guess this is maybe a good point to sort of bring up, and especially seeing as one of the studies that we've focused on, Gerry, has been your study on MERTK, one of the things that we've talked about. So, and I guess what you talked about there is exactly what you've gone through to sort of get to the point where you're at now, and other people have gone through, is that you identified that this was potentially a susceptibility gene, and then the process of looking into that protein and what that protein does, and we sort of mentioned in the summary a few little things that how MERTK may be involved in MS. But do you sort of want to talk about that process a little bit, and more so now that you're at this stage and you've identified this may be a susceptibility gene, we see a sort of potential as to how this protein may be involved. Where do you go next from there?
Yeah, sure. So basically, I guess the group firstly became interested in this family of proteins by using a microarray, looking at which genes, which expression of which genes were changed in an animal model of MS, the inflammatory one, EAE, and we saw that one of the families of proteins that were changed was this TAM family of proteins. So from there, we moved on to animal studies, looking at mice in which one of the genes encoding one of these proteins was knocked out. So these mice didn't have that particular protein, and then looked using one of the animal models of MS to see whether there were any differences between the mice that had the gene and mice that didn't. And there we found that mice that didn't have that particular gene, so this gene encodes for a protein that activates the TAM family of proteins. They displayed a worsened demyelination compared to mice that did have that protein, which suggests that it obviously plays a role in regulating the demyelination process. Subsequently to that, I guess I joined the lab, and I undertook a genetic association study looking to see whether any variations in those genes were associated with MS, and that was the paper that was summarized on the website earlier. And now, I guess now that we have found those associations, and also other people have found them independent of us, we are more interested in looking to see, especially in MERTK, whether deleting the MERTK gene from mice, whether that causes any differences during the demyelination process in those mice, as opposed to mice that do still have the MERTK gene. And also, some of the proteins that activate MERTK, for example, circulate around in the blood, and some of my work now is also looking at whether levels of those proteins are changed in the blood of MS patients or people with MS compared with people without, so healthy people. And what I found there is that one of the proteins that do activate the MERTK receptor I've seen that it's actually lower. The levels of that protein are lower in the blood of people with MS compared with normal, healthy controls. So I guess that can be a potential predictor or biomarker of disease activity, and we're currently trying to look to see whether that decreased level correlates with, for example, disease severity or duration or other such indicators as this.
Yeah. Thanks, Gerry. As a follow-up to sort of the polymorphisms in the iron-binding gene, Helen has asked a follow-up question asking if anyone's been following the research from Vanderbilt University regarding MS and iron metabolism and its impact on the immune aspect of the disease. Any of you up with those studies?
No, I can't say that I've come across that work.
No, I haven't, sorry.
Annika, have you?
No, I haven't. But I think it's interesting how different groups have a specific interest, and it's really good that they do because one group can't study everything. So, yeah, so it's good to know there are people looking into it because, as Helen pointed out, it's interesting associations and things that needs to be explored.
We'll look into that research, Helen, and we'll put a summary up on it over the coming weeks on our Facebook page to sort of look into that work. Tony, Brad, any questions from you guys for another 5-10 minutes?
Yeah, I was just a quick question. The genetic trials and research that's happening, the information of the actual MS people whose blood you're sourcing and using, are these anonymous research projects, like the patients are de-identified?
Absolutely,
I'm
yeah.
just wondering about any potential implications down the road.
Yeah, yes, they are definitely de-identified. But we obviously collect data such as sex, age, what kind of disease level they were at when their blood was collected, and so on and so forth. So anything that could help us, I guess, kind of draw any links to whether something we're looking at is a marker of disease progression or the current severity of disease. Or any other links potentially, but yes, the samples would definitely be de-identified.
Yes, so we tap into MS Base, which I think has been featured as well on MS Translate, and basically what we can see on MS Base is just the outcome data, so the MRI scan information, any clinical information associated with a number. So that's the only information that we have, and that number matches to the sample that we get. So only the clinicians and the nurses and people that are actually entering the information can see your personal information. We only receive like a number to work with. So that's the way that works.
Helen's just asked another question. She's wondering, where can a person with MS get genetic testing done in Australia, and especially Melbourne? Is that something that's being done at the moment that you guys know about?
Oh, well, as we don't really know what genes are involved in MS, it's really hard to do any sort of panel testing, so we don't really know what's important and what's not. So yeah, it is very hard. I know that there are companies over in America that offer some sort of genetic testing, and they include MS in that, but I'm not sure how much information you'd get from that. I mean, the types of studies that we're doing are really discovery studies to try and find out, what genes or variations are important and what's not. So until we sort of understand that a bit more, I think it would be really hard to do any sort of genetic testing for MS.
Yeah, there are definitely companies around in Melbourne at the moment that are starting to do genome and genetic testing, but as Melissa says, until we really know what genes are involved, how accurate they're going to be in terms of predicting any sort of MS progression or occurrence are quite low. Helen's followed it up with a comment saying, can they look at the genes involved in iron metabolism or genes involved in vitamin D metabolism?
Yeah, I mean they could potentially look at them, but I mean, I guess some of those variations would also be involved in other diseases and not specifically to MS. So I guess even if you did get those kinds of tests, it would be I guess, a really good predictor of risk of MS in the end, because as Mel said, we don't really have, that panel of genes that are really that we know are definitely important in MS. We only have some certain clues here and there, but we're not sure exactly what the exact genes to look at are at the moment.
Yeah, and I mean, even the genes that we do know at the moment, or that we sort of predict are susceptibility genes, they really just increase your risk of getting MS.
Exactly. Yeah.
It doesn't mean that if you have that gene or that form of the gene that you will get MS.
That's right.
That's exactly right, because the genes are unfortunately common in the general population. So making a link between why do I have MS, is it my environment, is it my genetics, which is Helena's next question, would be pretty much impossible to say definitively at this point. And I think anyone that says that you can do that is probably stealing your money, to be honest. But wouldn't it be, I think that's what all of this work is working towards, isn't it? Is to try and find a more individualized way to say why someone has MS. How will this person respond to treatments? Would they be better off doing something than, this treatment rather than something else? And that's, what I think, we all would want for the person sitting in front of us in a clinic, is to say, look, this is you, this is what's going to happen to you, and this is absolutely the right thing for you to do right now. And, but you have to dream big and, start small, I think.
Well, I guess the concept of the fact that there's environmental factors in MS as well is one of the most complicated things for people who are trying to do research into MS. And you guys might want to comment on this a little bit more, is that even for people who obviously might have a family history of MS that suggests that there's a genetic component to it. I don't think anyone questions that there is a genetic component to it. Obviously, families tend to congregate in the same sort of environmental situation as well. So how you can sort of, I don't know if you guys want to comment on that at all, as to how you sort of bring that together and sort of separate out the effects of those two factors.
Yeah, I mean, there's definitely a genetic component of MS, but it obviously doesn't explain the whole disease, and there's obviously a very strong environmental factor there too, because if you look at, I guess, twin studies of MS, the risk of one twin, when one twin has MS, the risk of the other one, the chance of the other one having it is only about 30%. So obviously that's not, that doesn't explain, genetics doesn't explain all of MS. So environment is also a very important factor, and I guess to try and dissect out some what those are for each individual person are very, very extremely difficult.
Yeah, I guess really the only thing that we can do is try to control as much of this in our experiments by sort of starting off in selected populations. So, targeting, for instance, Caucasians, living in Melbourne, their disease durations around the same time. So we do as much as we can to try and keep our populations, quite well controlled in our initial studies. But, it's difficult. I mean, without knowing what the environmental factors involved are, it's really hard to control for these things and tease these issues out. Yeah.
I think the common theme that's coming across is that MS is a very complicated disease.
Absolutely.
That are trying to look into it because of all of these factors. All right, we might just have final questions for everyone. If anyone's got a final question to ask, Tony and Brad, do you guys have a last question or comment that you'd like to make?
No, I think we're good.
Yeah.
Good.
Thank you very much for explaining your research.
Oh, you're welcome.
Thank you, no worries.
Annika, any final comments?
No, I think no, I don't have anything else to add.
Helen, any last comments, questions?
Helen was just commenting about how unlucky she's been. She says that she had two friends sitting next to her in primary school who both got MS, as did she, and they all have had different outcomes. So there has to be some environmental factor, you would think.
Yeah. Yep.
Okay, well, with that, we might bring an end to the broadcast, and I guess the final thing to say is thank you very much to both Gerry and Melissa for giving their time tonight to talk about their research. As always, it's much appreciated. We'll continue to follow their research with much interest and bring updates as they come to hand. But thank you again. If you'd like to stay on, we'll take a few more questions if anyone has them. All right, thanks everyone.
Thanks.